So the first step of meiosis is interphase. You have your nuclear membrane, and then you also have your spindle fibers and centrioles on the outside of it, and then on the inside you have chromatin. G1 is the first thing in interphase, and that is just the growth stage. So here you have the chromosome, and then you have half of the chromosome, which is a chromatid, and then you have the centromere, which connects the chromatids together. And then S is just the duplication stage. G2 follows S and is just what checks for errors. Following interphase, you have prophase 1. This is where Edwards syndrome happens, where there's a random addition of a chromosome. Also in prophase 1, the membrane disappears, mitotic spindle begins to form, and crossing over occurs. Crossing over is when chromatids of homologous chromosomes cross over and make new combinations, which causes variety in DNA. So next we have metaphase, which is where the chromosomes line up in the middle and completely attach to the spindle fibers. After that is anaphase, which is when the spindle fibers pull the chromosome pairs to the opposite ends of the cell. And then it's telophase, which is when the nuclear membrane forms and the cell begins to split. Finally, we have cytokinesis, which is when the two new cells are completely formed and separated. A cleavage furrow is what just happened, and it happens in animals. And then a cell plate happens in plant cell division, and it makes a new cell wall. Prophase 2, the membrane goes away and the centrioles duplicate. In metaphase 2, the chromosomes line up in the middle. In anaphase 2, the paired chromosomes get pulled apart. In telophase 2, the nuclear membrane begins to reform and they start to split. Cytokinesis 2 is when the 4 daughter cells are produced and you end up with your gametes.